going to continue in the sequence-based similarity section, which is module number 2. And whether you're doing this right after you did your blast or if you're doing this at a totally different time doesn't matter, okay? Because we're recording things in our notebook online here at GENI-ACT, you always have the ability to go back, which is nice. These are modules, so we can do them at different times. So you're going to log into GENI-ACT. I am, in my case, at 00010 notebook. And remember, this is a gene from Chidococcus sedentarius that's actually been studied, and I have a key in our files so that you can go and see what you should be writing down in a section about your gene from Clostridium. I can't do genes from Clostridium because they haven't been studied yet. So as a demo, this is a good one you can check against the key. So just remember I have that for you. So where we left off last time, you do need to go in order. We have our basic information all done. I'm not even going to go there. We have our sequence-based similarity data started, and we ran what was called a blast search. And what that gave us is we used 2 databases. One was a really highly curated database that has genes that have been studied in a lab and confirmed. And then we have what's called the NR database, which was just computers have determined that those genes probably exist, but they haven't, for the most part, been studied by a human at this point in history. And we recorded what the name of those top matches from each database were, along with the organism names and how well the alignments fit together. We even included a picture of what our gene compared to these high-quality matches were like in Chidococcus sedentarius. Then we did the same thing with the NR database because NR is a little bit lower quality than Swiss Prot because Swiss Prot genes have been studied in a lab. So you have more evidence that they actually do what they say they do. And we included our NR alignments down here. So now we're at the next section. And it's actually going to use the same blast information. So again, if you're here at a different time, that's not a big deal because all we have to do is grab some stuff and start again. So it says we're going to be clicking on the CDD search results at the top of the blast results page. If you're using the main manual, I'm on page 43 for this, and it's CDD stands for Conserved Domain Database Search. A conserved domain of a protein would be a section of a protein that seems to be represented in multiple different species, meaning it's been evolutionarily conserved. So it must have a useful function that through natural selection has been, shared from a common ancestor and preserved or conserved. So CDD is going to be using your blast search and looking for these specific sections of your protein that seem to be represented in other bacteria that are out there. I did a fairly quick CDD search just online, and I got a result that somebody did this study. You can see the citation here about coffee, and the way they show some different strains of coffee, the conserved domains are these C blue boxes, C1, C2, C3, C4, and C5. And they're basically just sections of a whole protein that have a particular structure that seems to be shared between these different strains. So what we are going to do is run a CDD search. So the first thing we're going to need to do is go back to BLAST if you don't have it open right now. The link is right here, and if you control-click, it will open a new tab. And if you remember, we did a protein blast, so I click that, and it says to enter our FASTA sequence, which we have already done. I'm back in my GENI-ACT notebook. I click basic information, and I'm going to scroll. I don't want the nucleotide sequence. I want the protein sequence. And remember, that's because 3 of these letters code for 1 amino acid, but there's not just one set of 3 DNA bases that codes for 1 amino acid. Sometimes there's 3 or 4 different nucleotide triplets that can code for the same amino acid. So we're removing all of that redundancy by just using amino acid sequence. And remember, each letter here stands for 1 amino acid. It's just a code. So I Control-C to copy. I go back to BLAST, and I'm going to paste that in, and I'm not going to touch anything else except to make sure that my FASTA header is up here. BLAST is going to ignore everything here, but that title is going to be important for me. Now with Clostridium, because some Clostridium genes have been researched, when you do your gene, you might want to exclude Clostridium, so you don't see just 50 billion Clostridium results. Clostridium's a genus of multiple bacteria. Okay, but there's a sheet about that in our files that you can look up if you notice that all your results are from Clostridium. Okay, but for this case-ed thing, I'm just going to run it so that you can get practice. So go ahead and run your blast. NR is fine. Swissprot would be fine as well, because this CDD is run whenever you click this blast button, and I'll show you where it is. We just didn't look at it yet. Okay, but they will be identical whether you use NR or Swissprot, so there's no point doing the 2 tabs and running it for the CDD part. That was for the blast part in general with the alignments. So I'm going to blast. With this in mind, I am going to pick Swissprot because it's a smaller database, and so my search will run a little faster. I ran an NR search a minute ago, and it was taking over a minute, so, you know, save yourself some time. So all I did, I kept my same FASTA sequence, and I picked Swissprot from the dropdown. And now I'm going to blast. And it took about 10 seconds, but here it is. Now before, we went right down here and we were looking at all these red things, and then we went to below that and we picked the low E values with a high coverage rate, and we picked 10 of those and we put those in a multiple alignment and saved it as an image, okay? But what we scrolled past is right up here. And what it lists up on the top here are putative conserved domains that have been detected. So this green and blue here are things that it's saying are part of a larger group of bacteria, and these things can be called COGs are one of them that we'll talk about, but superfamily is another one of those terms. You can see that if you look really closely, oh, there's a confirmed ATP binding site, okay, as a part of this protein. There's some other motifs, which are a section of a protein that have a specific function. An arginine finger, okay, that has a certain shape that's distinctive, okay? So you get the point. There's little domains, little sections of the protein that are known to do something. So what I'm going to do is just click on this graphic, and it brings that up in a lot more detail. Now, this is a lot of information. You're going to see that DNA-A thing that have been talking about already, but we're going to scroll down, and the only part that we want right now is going to be in your notebook. So I went back to my notebook, and I'm going to scroll back down to where we were. We're in the CDD section, the Conserved Domain Database, and it's asking for a COG number, and that's what we're looking for. So a COG is a cluster of orthologous groups. Here's a picture that illustrates the idea of an ortholog, and it goes back to evolution. So let's say we had a bacteria with a particular gene, and it just clones itself because bacteria are asexual reproducers. So it clones itself. Okay, if sometime along the way, you guys know DNA can mutate. Sometimes huge sections of a chromosome or a section of a chromosome can duplicate itself. Okay, so this is a duplication mutation, but it's still within the same species. So that's when it's called a paralog. That's where you seem to have 2 copies of a gene within the same bacterial species. It becomes an ortholog if speciation occurs, and that what once was a single bacteria later becomes 2 separate species that both have a similar gene in them from that common ancestor. So it's orthologous. If it's in different species, it's called a paralog, or paralogous if it's in the same species. So it's a way to describe these little particular groups or domains of a protein. And again, these domains that seem to have a well-established function are thought to have come from a common ancestor and been conserved. So I'm going to flip back here, and I'm going to look to see if I have a COG. And you can see there's a bunch of like PFAM and other things, but oh, there is a COG right here, COG 0593. So this is something I'm interested in right now. I'm not interested in the rest of it yet. So when I find a COG, you might have more than one when you do your Clostridium gene, and that's okay. You would just include both. I'm looking for my top hit, which in this case, for case-ed 00010, there's only one COG hit. So I'm going to click the link. I need a COG number. So I'm going to copy, Control C. Got to click the notebook to open up the editor. I tried copying and pasting, but because it's white text, it showed up as white in the notebook, so I just typed it, okay, and kept it simple. Please don't ever forget to save. So I've recorded the COG number. The next thing it needs is the COG name, which is right here, DNA-A, but I'm going to take the full name, chromosomal replication initiation ATPase DNA-A, and I'll just take that whole shebang and save it. Next thing I need is an e-value, and this is going to judge, again, just exactly how closely that matches my protein, in which case I have the KSED00010. That e-value, all I did is click the back arrow, and you can see, boom, there's the title. That e-value is 1.03 times 10 to the minus 153. Things are significant in this study if it's 1 times 10 to the minus 3. So this is much lower than that. And again, this e-value tells you what's the likelihood that that's due to chance. And when it's very low, it means it's an almost 0% chance that this match of KSED00010 to this COG 0593 is a chance. It's pretty likely due to conservation. So I paste my e-value and save. Now, if you have a second COG, you would do the same thing. But again, to confirm, I double check, there isn't a second hit. So instead of leaving blanks, which would later maybe confuse you and make you think you skipped something, I'm going to record this for myself. There is no second hit. And I'm even going to put not applicable in these other two blanks. And that brings us to the end of the conserved domains. There was a section of KSED00010 that has a sectional match to other proteins that have been discovered. And that's what that section tells me. I have some structure-based similarities. When you do your Clostridium gene, you may find it is very possible that there may not be a COG hit at all. In which case, that might mean that there are COGs out there that haven't been discovered that match your protein yet, or you just don't have one. Now, if that happens, you still record that. It would just be in the first hit, you would say, no significant COG hits found, and then I would continue and put not applicable for the rest of these, so that there were no blanks in your book. So that completes the CDD section.